From Microbe TV, this is TWiEVO, This Week in Evolution, episode 101, recorded on April 3rd, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent, great to be back together, live streaming. I think both of us might be cutting out our mics from time to time. We're doing some personal microbiology, some personal virology here with some head colds.
Just when I have to talk, my throat starts tickling me, right?
There you go. I'll drink to that. Cheers.
This is tea, by the way. Both of us have tea. It's not harder stuff.
Exactly. It's early enough in the day that the tea is
Although,
hopefully...
maybe hard stuff would help cut the phlegm, right?
Maybe. We'll see as the podcast unfolds what happens here. But
so now we're entering our second century here on TWiEVO.
Yeah, great to see you on the other side of TWiEVO 100,
Yep.
our live in vivo event in Johnson City, Texas. How was the rest of your tour of Texas, Vincent, after
Oh, it was great.
we were together?
I had, so that Sunday was the TWiEVO, and that was fabulous. You know, it was great. It was live streamed, and we got to see the museum and all that. And then Monday, Rich and I did a TWiV at the University of Texas with Jason McClellan. That's going to be released this weekend.
Very cool.
And then Tuesday, I did my little interview at South by Southwest with two individuals, and that was good. It was all good.
Glad to hear it.
Time. And then I went to some South by sessions the rest of the week. It was fun.
Oh, nice. Yep.
Have you ever been to South by?
I haven't. So that was the kind of the closest glancing blow for me, was being in town the same time that it was happening. I ended up leaving Tuesday morning to get back to work here at LD Lab Studios and Laboratories. And so, I haven't actually surfed in on the South by Southwest. What was really fun was after our live stream, our live event, someone who was participating in South by Southwest, drove from Austin to Johnson City to join us for dinner. And so we had a really fun barbecue dinner together and sort of celebrated that landmark of 100 episodes together.
He came to TWiV also on Monday.
Oh, very cool. Yeah. And so I think our hope is, to keep sort of tinkering and thinking about if it would make sense to connect at that time with the Science Mill and South by Southwest sometime in the future. So who knows what will grow out of these sort of seeds of science that we're carefully sowing.
Well, Nels, we're building spaces that celebrate science, right?
Yes, that's right. That's what we're up to here. Yeah,
it's. I'm happy to do anything that involves that. Let's welcome our little audience here. I'm going to ask you folks where you're from, so get ready. But in the meantime, I want to thank our moderators. We have Andrew from New Zealand. We have Barb Mack from the UK.
Fantastic.
We have Tom Steinberg from Eugene, Oregon. Let's see if there's anyone else here. Vanity Nutrition is here from Long Island. Les is here.
Great.
And that'll do it for our moderators. Thank you so much.
Yeah, welcome to our TWiEVO knots, our regulars.
Let's see who's here. Jan is from Tucson. Welcome. It's not sunny here at all. It's raining. It's been raining the last 2 days, and it's pretty cold. I'm still wearing hat and gloves outside. Peter's from London. M.E. Wheeler is from Helsinki. I'm going to be there in June in your fair country of Finland.
Wow. Will you actually be in Helsinki or whereabouts?
Actually, the meeting is north of Helsinki in a small university town.
Great.
I'm just thinking of, after the meeting, which ends on a Thursday, of going back and hanging out till Sunday. Fly back on Sunday.
Yeah.
So we'll see.
Idea.
Pete is from London. Welcome. Jeremy is from Seattle.
Welcome.
Lise is from Columbus, Ohio.
Oh, yeah. I heard there were tornadoes warnings or tornado issues potentially passing through yesterday. Hopefully everyone weathered that storm front that went through.
I have a switch here, Nels. It's called a
Yeah.
cough switch. It is aptly named.
Yeah, we're using it today. I think I have one as well on my mic. I haven't tested it out yet. We'll see where things
I've been
go.
fine all morning, and then suddenly when it's time to stream, I'm coughing. Jeez.
Well, yeah, I'm convinced the viruses want to be on the conversation. They're just saying hello to each other under the surface here.
Doreen is from Wayne, New Jersey.
Welcome.
Who else we have here? Ooh, EHFIK is from Vienna.
Great.
I guess you can see me coughing when I hit the cough switch.
Can't hear it though, and I'm happy to jump in where I can. Looks like a lot of spring weather across the country and around the world.
Gwen is from North Detroit.
Welcome.
Joseph is from Ontario. And we have our first contribution here of today from Rob.
Fantastic.
Thank you, Rob, for your support of science communication. Rob says, Seeds of Science.
There you go. Yeah, I really appreciate it. So the spring weather here is a little closer to that Tucson report that we saw a minute or so ago. We've got nice sunny weather warming up here. It really does feel like spring is on the menu.
Look, Mariah, is this correct? Mariah or Marija? I don't know if you pronounce the J. You tell me. From 55th Street. You're not very far from the incubator. You could come visit. We're on 30th Street.
There you go.
Nicola from Italy. Welcome.
Welcome. Good to see you.
BW Rock is from Sacramento, California. Vojta is from the Czech Republic. How cool.
Great. Very cool.
Nice. I like that. I like having a wide global audience, don't you, Nels?
Local to global, all the way from a few blocks from the incubator there to around the world. That's pretty fun.
Yeah, well, we have a long tail for everyone today, don't we, Nels?
Correct. 99 problems, but a tail ain't one, as the old saying.
Oh my gosh, this is spectacular.
It's gonna be really fun. Yes, for sure. And I think we're gonna have a lot of fun stepping through this paper today. And there have been some requests. Vincent, we've spent, as usual, a lot of time. I think we're drawn to the infectious microbes, given our own sort of scientific interests. But some requests out there to think about the evolution of our own species or our own closest relatives, the primates, including us. And so this paper came across the radar just in the last month or so. Really interesting one on the genetic basis of tail loss evolution in humans and apes.
Amazing.
Yeah.
Let me share this title up here. Let's see if I can get this to work. All right, here we go.
There we go. Published as an article in February, published online late February. So, we're about a month away from when this came out in Nature. It's open access. So everyone, if you want to take a deeper dive here, don't be shy about we'll post the link, but also you can go to Nature's website, even as we speak, and sort of play along at home on the podcast. So let's talk a little bit about the authors. First author, Bo Shua, and a big cast from NYU, New York University, speaking about the local neighborhood where you are, Vincent. Senior author is Ite Yane. And another familiar name, at least for me, is the second to last author, Jeff Buca, one of the giants of research on transposable elements, retrotransposons, and that will become that'll be part of our tale of tales today. So the Yanai lab is located in the Institute of Computational Medicine, the Institute of Systems Genetics, and the Department of Biochemistry and Molecular Pharmacology at NYU. Interesting side note here, Vincent. Ite is also the co-host of a great science podcast called Night Science. Have you come across
I have
this
not.
one yet?
No.
Yeah. So big recommend. We'll try to get the link in here as well. Great interviews. They've been running this podcast for a couple of years now, I think, and really trying to highlight or lift up the creative side of science. So night science is sort of this famous phrase. Do you remember who came up with this? The former Nobel laureate?
I forgot.
Day science, night science.
No. Oh, wait. Francois Jacob.
There you go. Yep, you got it. And so Jacob was thinking about how we operate as scientists, during the day, where everything's more formal, where we act scientific, sort of the more robotic or technical side, if you will. Whereas night science is where you let your imagination flow, you dream about ideas and sort of almost the impossible. And that it's in that confluence where a lot of interesting advances of science show up. And so it's sort of that idea of where creativity lives in the scientist's brain, and how bringing that in really marks a lot of the process that we don't always talk about during the sort of day science. Did you get your, the protocols exactly right? Are you weighing all of the variables? And so the sort of blend of art and science here. Big recommend for that podcast. And, I'd say good things happen to scientists that podcast, right, Vincent?
Yeah. I'm surprised I don't know of Itai. And, Itai is here at NYU, not far from the incubator.
Yeah, exactly. Maybe we could have a podcast fight or something, or a cross-pollinating interview. I don't know.
Well, we could bring Itai to the incubator and talk to Itai. That would be funny. That would be fun. I'd like
really
to
fun.
do that. I like cross-pollination.
We'll put that on our menu. So, but back to the topic of human evolution. And this is sort of one of those iconic traits, right? The tail, or lack of the tail. So let's set the table a little bit there. So I think the general idea is that our ancestors, and we're talking now, 25, 30, 40, 50 million years ago, all had tails. And the evidence of that.
What would they be? What would they be, our ancestors? Like mice?
Yeah, so they would, but this would now go back like 100 million years ago or longer to get to that common ancestor, maybe 150 million years.
Right.
Then things would be more like mice. Then you fast forward 100 million years, things are looking more like primates, right? So probably I'm doing a little maybe night science myself here, but like smaller kind of primate-like forms.
Yeah.
In all these cases, tails were sort of along the way. And so we infer that by the fact that all of our relatives, more distant relatives, in many cases, not every, but in many cases, have tails. And we're the ones, the apes, that split off from the rest of the primates, for example, the prosimians, 30 or so million years ago, and we don't have tails anymore. And so that's actually a way of really like kind of a pretty clear idea of when that was lost, how to date that, given that sort of sharp divide between the so-called hominids, and this is humans, chimpanzees, bonobos, gorillas, orangutans, and gibbons, our closest primate relatives. They don't have tails, and yet the African and South American monkeys do, and use them in ways that we'll sort of talk about. The sort of adaptive notion of why is it useful to have a tail. There's also a great.
So our most recent ancestor with a tail would be like an old world monkey, right?
Exactly right. And so, and then same with the New World monkeys, and also the prosimians, and then obviously mice have tails too, as another sort of mammalian example there. There have been independent losses of tails, including in some of the prosimians, so things like some of the loris species. There's also, again, if we look at some the domestication of cats and dogs, there's like breeds that lack tails. In some cases, that was the hand of human selective pressure or breeding pressure, where people wanted to specifically have breeds that lacked tails. And so there's all kinds of stories and a complex genetic, sort of story underneath the surface that probably has been written in the genomes in different ways for these different events.
Nels, I'm going through all my podcast hosts
That's great.
to grab your name. I started with Dixon. I don't know why. Maybe because you have a beard.
There you go.
So they're cats without tails, right? Manxes, famously, right?
Yeah, exactly. And
And
so,
they're fine. They don't have any problem balancing or running or anything like that, right? So maybe a
yeah,
cat, the tail is vestigial, not vestigial, but you just don't need it. It's just it looks good. People like to touch them, and cats don't like you to touch their tails, right?
yeah. So, and let's draw maybe the comparison here between the domesticated cat, which just needs to like convince its owner to feed it a couple times a day or something like that, versus a cheetah
Yeah,
that probably needs that tail to get up to that 60 mph. The sort of shape of the body, the form of the cheetah, but in order to counterbalance the weight and have that
huh.
stability when
So
you're
the tail is
running.
straight out, right, when they're running.
Yeah, I
Yeah.
don't know all the details, but that's not a trivial thing to be able to maintain that speed, to stop on a dime as you're like biting into a gazelle or whatever the prey situation is. The tail's not in that case is very much not a vestigial situation, and there's some clear adaptive reasons for maintaining a tail, or even the tail elongating or being sort of the morphology of it being very specific
Yeah.
to the lifestyle or the behaviors, especially as it relates to gaining energy for getting food, predator prey relationships in particular.
There are also prehensile tails, right?
Exactly. And so as we think about the South American monkeys, for example, right, as they're jumping around trees, they're navigating
Mm.
the canopies of the rainforest, having a tail with some dexterity is pretty important. What do we have here, Vincent?
I
What's
love this
packaging
mug.
your tea?
It says they call me neutrophil because I slay all day. I'm just showing this because Vanity, our moderator here, gave this to me a while ago. It's pretty cool,
Fantastic. There you
and
go.
it's got yeah.
Good.
I should show it off. Yeah.
That's a good way to get through a head cold. You're sort of advertising
Thank you,
what's
Vanity.
happening
Yeah,
immunologically there
actually,
as well.
that's what's happening. I got neutrophils acting up inside me. Yeah.
There you go. Truth in advertising here on TWiEVO. Um, so, yes, all kinds of these adaptations. And then so the question arises, why would you give this up? And so why don't humans have tails?
Mm.
That's a pretty conspicuous trait. And so, I think, again, kind of not controversial, but certainly not like a kind of, not a closed case either. But part of the thinking there is that when our ancestors moved from walking on all fours to being bipedal, or using standing up on our 2 feet,
Yeah.
that having
Yeah.
a tail was probably a little bit, not optimal in terms of the way we get around. Um, you can also imagine, scenarios where, as our ancestors were kind of picking mates, that tails were not so fashionable or something like that as well. Although the whole class, all of the anime, for example, or things like this, the tail sort of reemerges as a fashionable add-on, but
Yeah.
probably not
It's true.
kind of in our natural evolutionary history here in the last 30
You
million
have
years.
to modify your dress to wear a tail.
That's right. Yep. Tails don't come without their costs, and actually, that will be part of losing the tail. That's part of an interesting facet of this story as well.
But all the Homo sapiens and the Neanderthals and the Denisovans, they had lost their tails already many years before, right?
Correct. That's the idea, based on the fact that modern day chimpanzees, gorillas, out to gibbons,
I should say their
don't have.
ancestors lost the tail because they never had one.
Exactly. That's exactly right. And so as we date that, we're thinking about Neanderthals, et cetera, that's kind of on the scale of 50,000 years ago. And the sort of prevailing notion in the field is that this is more like 30 million years ago when these actual events were happening. The first events were happening. And that doesn't mean it sort of ends altogether, which is sort of an interesting conversation point here as well. And so what we have, Vincent, is actually the remnant, I guess you could say, in a sense, of the tail. So we have a tail bone, and this are 3 to 5 vertebrae. I think they're technically the caudal vertebrae.
Right.
And so, and that, how you talk about this is kind of important. So is this the complete loss of the tail, the retraction, or is there a remnant? Those are that's part of the mechanism here of what's going on here. So in that sense, you could make the case that the tailbone is just a super short tail, depending on how you want to think about that. So, we've known for a long time the morphology or the sort of form of our tailbone, but we don't know the genetics. How did we sort of step through, our ancestors step through this where it was lost? And so this is kind of like, a really great time to sort of dive in specifically to human evolution in some ways. So we have the power of genomics, getting, if anything, stronger by the month, stronger by the year, where as more and more high quality, well annotated genomes come online, including ones of modern primates, our closest relatives. And so chimpanzees, for instance, and bonobos, we infer that our last common ancestor was hanging out about 5 million years ago. We can start to ask or draw comparisons to ask, okay, what are the shared genetic changes or shared genetic features that you see among our closest primate relatives that differ from the ones that have a different trait, in this case, the ones that still have a tail. And so as you were pointing out, Vincent, that kind of draws the comparison point between the African or old world monkeys and the New World monkeys, the South American monkeys, to the hominoids. And then you can still go out the tree and compare that to things like mice and cats and dogs, et cetera. The comparisons get harder there because just the farther you go, or the more distant the relative, the more genetic changes, the more noise that you're weeding through to have that comparison with what is the, at least, or what are the changes that are candidates for encoding the differences in our tail development, in our tail morphology. So what genes changed? And so the authors, I think, really nicely kind of emerging as experts in sort of genome hunting, of trolling through genomes, making comparisons, and then trying to pull out ones or work on ones, do experiments to try to gain evidence for whether those changes that they identify in the comparisons actually do something functionally for the difference in tail development, or in this case, the quote unquote loss of the tail. And it's tricky. So it's kind of a balancing act because you can't just, again, look at every single gene. There's lots of them, or all of the genomic space, because the number of changes just very quickly build into the millions, tens of millions, and so you really need ways to prioritize. And so here the authors take advantage of decades worth of developmental genetics, which is also advanced. In particular, the mouse model. So mice have tails. There's natural variation in tail length. There's all kinds of developmental biology. So there's a knowledge base for exactly at what moment in developmental time does the tail start to develop? Does it specialize or differentiate from the other tissues that are developing around there, even the other vertebrae that contribute to the spinal cord? And so, from some of these data sets that have been put together and curated in useful ways, there's something like 100 genes that have either been mutated and or there's natural variation that's been identified that influence or impact tail development. And so that's an obvious place, right, to kind of move the comparisons from the primates instead of across the whole genome. Let's look at these 100 genes or so in the genetic regions that might be controlling the expression of those genes. When do those genes get turned on during development? How much of the gene to make the protein that then is doing all of the work of the developmental biology here.
So Nels, can I step back just a bit? So this paper basically is to try and get insight into what kind of, the mechanism of tail loss, and then ultimately the selection that would lead to that. Why do we care, Nels?
Yeah. Yeah. For other than sort of cosmetic reasons, right? So, so I think it's, so I think the mechanism is, it's complicated, but in some ways it's more accessible. All of the getting at the, the why question of why was the tail lost, or what were the selective pressures, that's a little more challenging. I mean, there's some obvious inferences as it relates to the behaviors we've been talking about, getting around in trees and sort of letting go of that, the mechanics of moving on 2 feet. Why is this important? I think, it's kind of like, maybe I'll draw on the senior author's podcast, Night Science. Like, if we let our kind of imagination roam a little bit, there's something pretty just, I'm thinking about growing up and reading, kids' books like Where Are the Wild Things and all of those monsters that have tails. There's something in our human, sort of psychology almost, where the tail is a pretty iconic structure as we sort of think about ourselves and the world, or the animals and monsters around us. And so, identifying that, or capturing our imagination, but then using it as a model to understand what are the mechanics about how genomes change that can influence these sort of core developmental traits. There's probably some rules in there about how this process not only plays out for the tail, but all kinds of other structures. Why is it that we have 5 fingers on our hands, 5 toes on our feet, right? As we start to learn the mechanisms, that might give us the tools to then sort of recast all of the other developmental stuff that's going on, to get closer to a more full understanding there. And then why do we care about?
I want 6. I want 6 fingers now.
Yeah, so we could go. That exists polydactyly among, for example, Hemingway's cats still walking around in Key West, Florida. But there's also, on the flip side of the coin, in addition to that, sort of genetic fashion show that you're describing, or how we sort of select on the animals, cats, dogs, et cetera, around us, there's also human disease. A lot of diseases involve errors in development, you could say, that have some pretty strong consequences in terms of how individuals of our own species grow up, or how they live their lives. And so as we get closer to an understanding of what can go kind of off the rails developmentally with some of this knowledge, that could be really valuable for interventions of the future that could, help people to be able to operate in the world with more ability than you see in some cases, given all kinds of genetic diseases and other related sort of human biomedical conditions.
I would also argue that so many pathways and developmental pathways overlap.
Yeah.
Like transcription factors can do different things. And so if you identify how a tail is lost, you could also see, why didn't this happen? Why didn't we have this other effect, which obviously could be lethal? Because then you start to see all the networks that participate and how changes can get around that. I think that's really important too.
I couldn't agree more. And also just the complexity of this, right? So when are genes turned on? Is it the regulation of the genes? Is it different? Is it a mutation or a difference in the gene itself? And can that teach us about the function of how these transcription factors, for example, work together in concert? It gets complicated in a hurry. And so I think one of the big challenges to really sort of pull this apart, or take this kind of reductionist approach, is to have clean phenotypes, to have sort of the ability to measure something. So you make a genetic change or observe a genetic change in nature and try to link that to a very clear functional difference,
right,
along those lines. And the tail is like, it's great. So because you can quantify it. You have one or you don't, yes or no, or you have this many vertebrae, it's longer or it's shorter. It's very easy to measure sort of an outcome or a phenotype. It's a great model system for gaining this knowledge. And in addition, so the mouse model, sort of musculus, the house mouse, has been a workhorse for developmental studies like this. But there's also, other work in natural populations. In another paper, which we could have picked just as easily as this one. So it's not about humans, but it's about deer mice. This is a new paper from Hopi Hoekstra's lab. She's been on TWiEVO before. And so they were looking at deer mice. And the paper that just came out, I think a week after this one, talks about tail length adaptation. And this connects tail development to this very natural evolutionary process. And so in this case, the deer mice, there are these like different individuals in the population, some of which are the forest mice versus the prairie mice. And the forest mice have longer tails. They're climbing around on trees. And so this other study links that difference in tail length and the notion that it's adaptive if you're a forest mouse to have a longer tail because you can climb better, to a variation in a gene called HOXD13. And so we're going to get a little bit into the world of alphabet soup for gene names here. So HOXD13, this is part of this sort of really well-described family of genes, the HOX genes, which are involved in sort of the vertebrate body plan, segments of the body. And so adding or subtracting vertebrae here can lead to tail length, differences in tail length, different than the presence absence, but it's a very natural system. Also connects it, kind of immediately connects the dots to the behavior as you differentiate these phenotypes from the lifestyles of these mice, the ones that are hanging out in trees versus the ones in the prairie that have shorter tails. So in that study from the Hoekstra lab, they went to or saw differences in this gene HOXD. For this study, they end up zooming in on another single gene, a different transcription factor called TBXT. And so again, classically studied gene for early developmental processes. And so, that kind of gives it that priority access, in a sense, to being something that you would want to make draw comparisons between primates that have tails or don't. It's a sort of very natural candidate region. Other changes they see, and they kind of don't study that here, but are probably very much, the authors aren't trying to make the case that this is the only genetic change underlying this, or the only genetic change in town. Wnt 3A, MSGN1, or some other of these alphabet soup gene names where they see interesting differences, but decide to really focus on this difference in TBXT.
So TBXT has previously been associated with differences in tail length in mice, right? So that's why they kind of zoomed in on it, correct?
Exactly right. And so that's what comes from using that mouse model. So among those 100 or so genes or mutants are described. So you can query this database for phrases or keywords such as absent tail or short tail or vestigial tail. And so geneticists in past studies have, observed mice without tails, and then they've linked it to some changes in the mouse system. So what the authors in essence are trying to do is sort of port over some of that knowledge from mouse to primates and see if there are newly described differences here that could be could be explaining some of this evolutionary biology. So the authors do this kind of gene gazing or observations, and in the end, they ended up comparing something like 140 genes that they had sort of prioritized from that mouse database. And what they're looking for is shared mutations that are found in the apes but are absent in other primates, with the idea that there was a change or mutation in the ancestors of all of the apes that went on to all the modern apes that differentiates it, that helps explain the loss of the tail. And again, they found, hundreds of differences, thousands of differences even. And so this is where maybe another kind of call out to the Ite's, the senior author's podcast, Night Science. How do you narrow down and start? You can't test everything. And so how do you make choices here? And I think this is really, in some ways where evolutionary genetics or genomics is kind of a little bit more of an art than a science. You're kind of using your intuition in some ways, and you're really kind of drawing together a lot of knowledge to be creative, to think about ways of deciding what you're gonna test. And the reason you need to be creative is the work that goes into really testing this. And we'll step through this a little bit together here in the next several minutes. These are like years of and into the hundreds of thousands of dollars of investments to try to chase after some of these changes to see if there's evidence that those genetic changes could actually influence the absence of a tail in us and our closest primate relatives. Okay, so Vincent, it doesn't take us long or we can't sort of escape our microbial interests here, right? So as they were going through all of these changes, excuse me, one that really sung out to them was the insertion of an ALU element in that gene, in the intron of a gene TBXT. So let's unpack this for a minute. So ALUs are these, it's a form of a transposable element, a remnant in a sense of, or a non-autonomous, I should say, retrotransposon.
Do you know why it's called ALU? ALU, Nels? Do you know why?
I don't remember. Help remind me.
You're too young to know. So I worked on ALU elements ages ago. So it's because it is, if you cut with the restriction endonuclease ALU1, which comes from Arthrobacter luteus, it's a restriction enzyme that cuts at a specific sequence. That liberates this element, which they found was highly repeated in the genome, present about 1 every 5,000 bases or so. So every gene is near some ALU element of some kind. And so it was originally defined by that restriction enzyme, and it turns out to be what is it? It's a retrotransposon? Or is it?
It is. Yeah, it is, exactly. And so, but it's non-autonomous. So these are also, again, we're kind of an alphabet soup here, but also called signs or short interspersed nuclear element. Right. And so it's a cousin to the lines or long interspersed nuclear elements. The lines are autonomous, so they encode everything they need to copy and paste themselves in genomes. And then the signs, including the ALUs, are hitchhiking a ride, basically. They have enough of the genetic identity to the autonomous elements that they can convince those enzymes, or the enzymes recognize that sequence and copy and paste it throughout the genome. And so, so
let me diverge just a minute here.
Yeah.
So ALUs are very common. One of the most highly repetitive elements in the human genome, right?
Mm-hmm.
In fact, it's likely that there's an ALU next to every gene. It's so frequent. So we took advantage of that when we were trying to clone the human poliovirus receptor.
Mm-hmm.
Because what we did is we took human DNA and broke it up and put it into mouse cells, and we identified mouse cells that could bind poliovirus, right?
Yeah.
But how do we pull the gene out? We don't have any probes, hybridization probes or anything to pull it out. This is pre-genome sequencing, right?
Yeah.
So we thought, let's use a radioactive probe derived from ALU element, basically. So it has been cloned for ages in plasmids. So we took it, we labeled it up, and used it to hybridize to colonies and identify those cells that had candidate human genes, right? And you do that a few times, and eventually you get it. So in the absence of a gene-specific probe, you can use something that's bound to be next to your gene. It's just really cool.
Yeah, I love this. I also just love how this kind of echoes through, right? You're using this sort of virus-like sequence. I call them ALUs. I think I've been living by Alta Ski Resort, and so I have my pronunciation here for ALU versus ALU.
But we used to call it ALU, yeah.
Yeah, we're talking about the same thing. But, so it's this virus-like element, as you're saying, that's sprinkled throughout the genome. You're using it as a marker to get at a gene that's the receptor for a virus. And so it's almost like these echoes, right, of the viruses, and the virus are helping us technically, or teaching us, or we're using these as tools. I mean, this is the motion of evolution. It's all kind of written by viruses and virus-like elements, and then all of the complexity that sort of builds out on top of that. And so, just as you're saying, Vincent, these things are super abundant in genomes. So like a quarter of the human genome is devoted to these line 1 elements, the autonomous cousin of the ALU, of the ALU. And
I got you. You
and then another.
Yeah.
No, I'm not feeling. I'll say it probably both ways. And so, and then the signs take up another like massive chunk of our genomes as well. And and so the genes that encode the proteins are only like 3%. So the genes that we think of that, basically encode the structure, the proteins that make us, are only are like in a huge minority from sort of the genomic real estate that's dominated by these virus-like elements. And so of course they're going to have a hand in our own evolution. And I think that's one of the, real sort of foundational points here. This is a sub-theme of TWiEVO, is that we try to think about humans or ourselves and sort of this level of evolution. And pretty soon, as you peel back the layers of the onion, you're going to find a virus. You're going to find a virus-like element or retrotransposon. You just can't. That's who we are. And so, I think that's one of the things I love about this paper. It sort of reminds us of how genetic variation sort of unfolds. It's really these infectious microbes or the related sequences that have driven the conversation, stretching back even billions of years.
So, Nels, how would you pronounce the restriction enzyme BGL1? How would you pronounce that?
Bagel 1.
Bagel.
Yeah.
But there are many people outside of New York who used to pronounce it Biggle 1. It's Bagel 1.
Yeah.
It's a bacterium.
That's right.
Bacillus something.
Yeah.
But if you're from New York, you pronounce it Bagel 1.
Of course. And I think most reasonable molecular biologists also pronounce it Bagel 1. That's another really cool connection here, right? Is so the restriction enzymes, which are part of the immune systems of bacteria, in this case are, named for the bacteria that has that cutting site recognition that happens to liberate these virus-like sequences, the ALUs. So yeah, it's all kind of this crazy web of microbiology that then we're sort of standing on the shoulders of microbes, you could say. Okay, so let's go back to TBXT. And so this, again, this isn't just, so the notion that TBXT is involved isn't new, but what's new is that insertion of a transposable element that could be messing basically with the function of TBXT, specifically in apes. Yeah.
So they find this ALU element in the hominoid TBXT gene in an intron, right?
Yes.
So, Nels, if you saw that, would you just pass over it because you said, that's not going to do anything?
Well, I wouldn't because I'm a big fan of retrotransposons, but most people would, and and in particular because of its location, just like you're saying, Vincent, it's in an intron, meaning that as the gene is being transcribed, so as we go from DNA to RNA to protein, that intermediate step,
Right.
that RNA, the introns are clipped out or spliced out, and the exons glued together to then make the final product of the protein through translation. So we're going through transcription and translation. And so, yeah, so that immediately you might down prioritize this because it's not going to be part of the gene in the end. But how introns are regulated, how they're spliced in or out, does could matter. And in most cases, so they actually, the authors spend some time on this in their manuscript. They say, you might immediately think this isn't important because it sits in the middle of an intron, meaning it's probably itself not messing with the splice sites, so to speak. It's not messing that up. And so it would be sort of inert. But what they noticed nearby here, and this is where it gets to that kind of the rules of what to look for that might be interesting, is there's another element, an ancient one, an older one, ALU element, that's sitting in the next intron over, sort of to the left. So the specific one to apes is between exon 6 and 7. The ancient one is between 5 and 6. And so what's also important here, a little technical detail, it's flipped in the other direction from the young one. And so there's now this sequence homology. As you go through that RNA intermediate, there's complementary sequence that would pop out exon 6 and potentially in what they call a back splicing event could eliminate exon 6 from the transcript, get translated into a protein that's a little bit shorter. It's missing some of that genetic information. And so that's sort of the candidate mechanism that they're looking at here.
And also, of course, it's hominoid specific.
Correct.
This ALU. It's in a gene that's known to control tail length, right?
That's right.
It's near this other ALU element. And the insertion time of this ALU corresponds to roughly 25 million years ago when the hominids lost their tails.
Correct. Yeah. So, this is all compelling circumstantial evidence that this could be part of the story of how our ancestors lost their tails or tail development changed in pretty substantial ways. So that's circumstantial. What you would love to do is to test this, right? To get some direct evidence or get different lines of evidence and do some experiments.
Can we do that in people, Nels?
No, certainly not in the red states. Probably not in the blue states either.
No.
I'm being a little facetious here.
It's politically independent. Yeah, it's not red COVID.
Correct. No. This is morally untenable to do these experiments on ourselves. And yet, here's where that mouse model system comes in again. So this is, one of the reasons that my colleague just a floor below me here, Mario Capecchi, was a co-winner of the Nobel Prize. Was he and others helped to develop the genetic tools to change genes to do so-called knockout mice, where you alter genes in mice, and then because we share so much of the same developmental biology, the idea is that we can learn about ourselves by not experimenting on ourselves, but to actually take this comparison point. So that's what the authors do here. They sort of lean in on that tried and true approach using some updated gene editing techniques. And actually, I should say, so before we get to the mice, they don't do the experiments in humans, but what they do is they do observe what's going on in human embryonic stem cells. So, for many reasons, scientists have established cell lines, embryonic cell lines from human tissue. And this has been massively important for advancing things all the way from in vitro fertilization to just many sort of core biomedical advances, is the ability to work with some of our own cells, but not grow this into actual humans. And so what they can do immediately is to ask, so far they see that we got excited about that arrangement of the ALU elements, but what you'd predict is that, given that these are early embryonic cells, that as those gene TBXT is expressed, you should see both the full form that includes exon 6 and then the other form that's missing exon 6. And so they actually design probes to amplify those genes, run it on a gel, and you can see the presence of that just by a difference in size, the presence of that TBXT transcript that is actually missing exon 6. So that's starting to get closer to seeing a real functional effect here. It's not just the presence of this insertional mutagenesis, the ALU element, but it's also the fact that you see the alteration of the gene itself from human cells. So then they go ahead and they actually use gene editing. So they use CRISPR. I think we've talked about this on TWiEVO before. So this is CRISPR borrowing again from bacterial immune systems. That's a very sort of tried and true theme over decades of molecular biology as we learn about how bacteria fight off the viruses, the phages. Within those mechanisms are these like really useful tools that we can use then to mess with genomes. And so CRISPR is one of these. It's a sort of been called the adaptive immune system of bacteria. We've labs like Jennifer Doudna's have repurposed this to use as a gene editing tool. So what they do is they use this to take out exon 6 and see if that matches, matches the loss of that exon. So yeah, here we can actually, it's hard to
show
explain.
You the picture. Yeah.
Better to see.
This is really great. This gene diagram here at the top, right?
I agree. So
that's the TBXT gene, right? The whole thing with all the exons and introns.
Yeah, the rainbow colors there. Each color of the rainbow is a different exon. And it's between the blue and the purple where they've drawn in a red element. So what they want to do is clip that out. They want to clip out the ALU because the prediction would be without that ALU there. So and by the way, that's kind of, that's reproducing what most primates look like outside of the hominoids, all of the African and South American monkeys, even in the TBXT gene of mice, there's no little red element. And so the scissors is how they diagram the CRISPR editing they do.
Right.
And then what we're looking at below this, that black and white film, those are the gene fragments. And so the first thing you see on the left two lanes, as we're working from left to right, are the top band. That's the full length TBXT gene.
Yep, right there.
And then right beneath it, that's the new transcript where exon 6 is missing. So in addition to the size, they can size it, they can also sequence it and pinpoint that sequence is missing, the exon 6, that dark blue box. And then what we're looking at then, if we go to the far right, so as the last four lanes, now that that shorter gene fragment, or the one that lacks exon 6, doesn't show up. If they remove that ALU, then it's just the full length gene. So the rest of that, what remains of the intron, is spliced out normally, and exon 6 is always included. And so that's a really nice piece of evidence that says, yeah, that ALU, that red ALU in the diagram really matters for the appearance of this different form of TBXT.
And they clip out the red, which is the one that was inserted more recently into the prosimian genome, right?
Exactly right.
And then they also nip out the ancient one separately because it should have a similar effect. Right? It should phenocopy the effect of taking out the exon 6 ALU.
Exactly. So that gets into that mechanism of that overlap. And again, so those are those middle four bands right below where basically you don't see that slightly shorter band. And then what they note though, so like, I don't know that there's they've edited this in a way that you don't actually see this in nature, but this sort of causes a cryptic, even shorter band to show up if you just take out the black one. And so then the red one, the more recent insertion of the retrotransposon, that probably is now somehow interacting with a different sequence nearby. So if it happened in that order, you might also get to sort of an effect like this. But that's sort of getting a little bit into the weeds of the details here. But this is great. So, not only do they see this candidate insertion, but if they mess with it, that actually influences the gene itself. So far, so good. They're kind of starting to narrow in on this. And so then what they would really like to do is to see if this matters for the phenotype. What happens to the tail? And so here's where you can't really do these experiments in humans or try to like basically clip that out, try to recreate a tail. And so what you do is the reverse experiment, and you do it in mice. So you use that same tool, the CRISPR editing. And by the way, so it turns out the TBXT genes, just as some background here, are between mouse and human are 91% identical. So we shared this as a common ancestor. We've diverged in some ways. We already know that there are some mutants out there where there are tail phenotypes. So what they want to do for the first time is make these specific changes, and ask whether if you get into that context, does this influence, or if you sort of phenocopy or genocopy in this case, because it's not an exact mutation, the same mutation here is technically hard to do, whether this leads to tail phenotypes. So what they decided to do is it's easy. So it's harder to put an ALU element in there. It's easier to just take away exon 6, right? Mm-hmm. Which would be basically the functional outcome of having the insertion there as well. So they do that. They use CRISPR to gene edit, to delete that. And then and they do that in one copy. So it turns out you have 2 copies of our genes, right? One from in this case, our mouse mother, one from our mouse father. and so they just mess with one of them and leave the other intact. And that also kind of genocopies the human case, right? So you still have some of that full length TBXT. And you just have like less of that altered form. And so the notion here is that it's that combination of the 2 forms that is sort of influencing the developmental biology here. So they kind of they come close, or sort of genocopy that by deleting exon 6. And then that allows them to then ask, does that matter for the developing mouse tail? And the answer was yes. Not 100% of the time, but in 1 out of 3 of these offspring that were missing exon 6 from one copy of their TBXT gene, they have tail phenotypes, meaning there's either the tails are short or there's no tail at all.
You got to look at this. This is just what I'm talking about. This is spectacular, right? I mean,
love it.
A mouse tail is really easy to score, right? Mm-hmm. And normal tail, there it is. And look, these mice that have short tails or intermediate length.
That's right. Yep. And so that's kind of that jackpot moment, right? Where you're in the lab and you're like, okay, we prioritize this candidate gene. We see this really interesting mechanism of how a jumping gene or a selfish genetic element is hopping around the genome that might be influencing this in a very specific way. But as we're kind of saying here, right, like this, and to their credit, so like, I think a lot of scientists would stop here. Like I would be tempted to stop here, right? Like, pretty close. Like we messed with this one exon, sort of genocopies or comes close, but they really actually go to the extra mile here. And things get a little more messy, but to their credit, they really want to get as close as they can to not just sort of approximate, but to get the genetics kind of to recreate almost, what was going on here with genetics. So they actually go ahead, and the gene editing technology is getting more powerful, or you're able to make more, not only take away genes, knock out genes or portions of genes, but to introduce genetic material. And so their goal was to introduce the ALUs into the same parts of the TBX gene introns and see if the mice lose their tail. Basically recreating, what we think happened 35 million years ago, sort of in the laboratory today, as if this happened like in an ancestor of mice, would all mice not have tail kind of energy? So some complicated genetic gymnastics, if you will. And because it's harder to put things in than take them out, they didn't quite get all of the exact genetics they wanted to, but they came close. So they had one line that added an ALU at the right position in the between after exon 6 and before 7. And then they had another line that has some reverse complementing sequence, but not exactly, it doesn't sort of totally match the ancestral or the more ancient ALU element that was upstream of exon 6 between 5 and 6. But they take these 2 strains, one that had the extra ALU, the other one that has some reverse complementing sequence, kind of approximates the ancient ALU, and mated them together. And 10% of those mice had shorter tails. So definitely consistent with their idea, but not as quite, maybe not quite as dramatic or striking as what you were just showing on the screen, where they went all the way from full tails and down to, in some cases, in their one third of the mice with phenotypes, with up to no tails at all. But now that they have these, mouse lines that more closely approximate the genetics of what we see in primates, they can really do a deep dive. So they can isolate these cells from the right embryonic time point when tails are just starting to develop and measure the amount of the different TBXT forms. And they start to see really they're sort of phenocopying the amount of the full length TBXT with the transcript levels of the one that had, or the form, I should say, of the TBXT that's a little bit, it's missing that chunk of exon 6, that chunk of the gene, and can work with these so-called heterozygous deletion mice, which are approximating basically what they think happened. And so they do this. They use their ALU mice, and they mate that now. Actually, they go to they kind of got into the things got complicated with their reverse complementing sequence. So they just go back to that exon deletion 6 mouse, and they mate that to their new ALU mouse. And then look at tails, and it gets closer to what you would expect sort of for the tail loss phenotypes. It's more apparent. So a lot of gets more complicated here, but basically it's all still pointing in the same direction, in the idea that the abundance of those different transcripts, both the full length and then, in some of that shorter version, is what's important here for tail loss. It's not just if you lose it all together, that's a problem, or that sort of explains it. In fact, that turns out to be a problem. So if you lose TBX, so you still have, if you just only have the short form of TBXT, so if both copies, the one from the mother and the father, are lacking exon 6, those mice don't survive. Actually, they can't develop properly. Around the time that the tail development is happening, all of development goes off the rails, and they have all kinds of spinal cord problems. It's actually lethal. And so this is really important, Vincent, because TBXT isn't only involved in development of the tail; it's entangled in the development of the vertebrate body plan,
yep.
of neural tube, et cetera. And so this raises a really important point, which is, let's say that the loss of the tail in our ancestors was adaptive. We were standing on our 2 feet, having a tail was kind of getting in the way. There's probably a cost associated with this, right? So ALU insertion, this is random, just, a selfish genetic element, cutting and pasting, propagating through a genome.
Yep.
Wasn't sort of designed with that sort of CRISPR technology or gene editing technology, which itself is still pretty sloppy. But these aren't precise engineering sort of solutions. And so you kind of get the good with the bad. So the good might be that our ancestors without tails were walking around a little more casually, or hanging out, or having more kids, et cetera. But the cost, and the authors point this out, is that humans, just as a matter of regular course, 1 in 1,000 human births involves neural tube closure issues. And so that might be an echo of this evolution. So the loss of the tail was adaptive enough that this trade-off, or this sort of increase in probability that you might have a developmental problem as it relates to the development of spinal cord or neural tube, could be sort of a consequence of evolution unfolding. This is really important because it really illustrates, evolution is not this super efficient, optimized scenario that the way an engineer would design developmental biology or would design a genome. It's random chance, mutations, changes in the genome, and then natural selection acting, not for the optimal form, but the one that's just a little bit better than the other forms out there, where the genes go on as it echoes through the generations.
So in this case, if you just take out both copies, it's lethal, right?
Correct. Yeah.
So randomly, maybe at some point, both copies got disabled, and that organism never went anywhere, because I'm sure it happened, right? Because that's the way random mutations are. But then you had one and one only, and maybe it shortened the tail initially, like you said, right? and then maybe some other changes come in and
Yes.
get rid of it, because it doesn't happen all at once typically, right?
No, exactly. And so then you can imagine, a case where, so in this case, even if you have, so the difference between removing exon 6, just in the mechanism that they did in the mice, and those mice, if you mate the two heterozygotes together to the homozygote, you just don't recover those mice. They don't live.
Yep.
In humans, it's different, right? So you can have both copies of that ALU. You still get the full length, but then from both copies, some of it is shorter. So you still have the full length because it's not physically deleted. It's just a sort of 9 out of 10 will still have the exon 6, whereas it's that 1 in 10 won't because of that new complementary ALU that's in there. So that allows you to sidestep that lethal situation, at least for that mutation, but it can still have these side effects. And so what you imagine is, there was enough ability. So clearly our ancestors could tolerate the presence of that new ALU insertion. But it probably wasn't optimal. And then you could have, since that arose, you can imagine compensatory mutations, both in TBXT, but in other genes that are involved in this developmental process that would mean that it was, some of those defects, there might have been, if we could roll back the clock or get in a time machine to like 20 million years ago, there could be a lot more live births with neural tube defects. And then that's selected on the ones that sort of were able to survive that new mutation. Those then continue to propagate. And so then you have sort of compensatory evolution where it actually gets more well-behaved. And we could still be in that process now. So if we came back in 30 million years, maybe there are more changes that get selected where it's even less likely that you'll have neural tube defects. I think, that gets a little more complicated, but just it's useful as a thought experiment to think about how the evolutionary process is unfolding.
And so then a thought experiment, if we could put the ALU element back into us, probably wouldn't make a tail because other things have happened since then.
Yeah. So if you remove that ALU element, right? So then you would basically have 100% of the long TBXT form that would match the case that you see in the old world monkeys, in the new world monkeys, in the mice, wild type mice. Then the prediction would be if you removed that, if we edited that out of our genomes, that all of a sudden we would see a tail. I think that's unlikely. Maybe, I don't know. I mean, so we have 3 to 5 of these vertebrae. Would you have 6 or 7? I don't, more common like that? Or are there enough changes or other sort of parallel mechanisms where you still wouldn't have a tail?
Well, they say they don't think that if you put the ALU-Y in, it would make a tail because other changes have happened over the time that it enhanced the tail loss or whatever. And so now that ALU may have started it, but it's not going to reverse it. Yeah.
Correct. Yeah. So if you took it back out, which would be the ancestral state, don't expect. So if you're doing like DIY genetics on your own, like obviously you can't do that. You're already past that developmental. There's no pathway. You're past that developmental window. There's no way to get a tail here, even if you could, or if you want your kids to have a tail, don't mess with trying to get rid of that ALU. It's probably not going to do the job for you.
So they make an interesting comment at the end of the discussion. They say, we don't know what the pressure was in hominids to lose the tail, right?
That's right.
Maybe involved in locomotion, who knows, right? But they say the selection must have been strong because there's a neural tube defect trade-off, right? As we've seen in some of these experiments. This transcription factor controls neural tube development. So if you need something, if it's really important, you're going to find ways to get around it. And we saw how that happens here to do just one copy, for example.
That's right. Yep. Yeah, and I mean, you'd love to know. So currently in live human births, I think 1 in 1,000 have a neural tube defect. If that ALU wasn't there, would it be 1 in 10,000? We don't know. That's complicated biology, but that's where the idea of the trade-off comes. The reason I think this is a really important, discovery, really important observation, is that and we hinted at this before, is that so the mechanism here, what they're calling back splicing, is we've got, as you're pointing out, Vincent, right? You've got ALUs just sprinkled all over the genome. If a new one lands in that reverse complementary orientation nearby another one, that could have this effect of messing with the isoforms or the forms of the genes that are translated, and that could be sort of impactful genetic variation. And no one has really put that together yet. That's really one of the key fundamental advances of this work. So now that they've seen this, that they've spent the years of time to build these mice to connect it to the phenotype, to see the loss of the tail, that sort of adds weight to, as we're scanning genomes into the future, look for those kind of arrangements, right? Use AI or teach your algorithms to pull out those patterns where two alleles are facing in opposite directions and are within, a KB and flanking some important genetic element, some important exon or other regulatory sequence, et cetera. There's probably all kinds of cases like this hiding out that could help us better understand the impact of genetic variation on all kinds of different traits, tails. Just go down the list of important biology, and that could be going on here.
Hey, Nels, I just figured out the selection for not having a tail.
Uh-oh.
So I realized the clothing industry wanted men to be able to wear pants, so can't have tails.
Yeah, I don't know what the tailors of 20 million years ago looked like. That's a high wire act, but so that's where I don't think we're going to be able to get to we can speculate on the why question, right? But you can also, the null hypothesis would be that, if you had a tail or not, it probably didn't really matter. But this is also who lived, and maybe there's other biology. The loss of the tail could be hitchhiking with other important sort of adaptations, as the hominoids diverged from the other monkeys that retain tails. All kinds of reasons to think why up to today modern monkeys have tails as we see them behave and go around the treetops, etc. Here's where,
but it's all quadrupeds, right?
Mm-hmm. Correct.
We're the only ones that are on two legs, right?
Yep.
The
That's
hominoids,
right. Yep.
And we don't have tails, so somehow related to being on two legs, most likely. But you may be right; there may be other things involved as well.
Yeah, that's a clear correlation here, and given certainly it's an attractive hypothesis. However, there has been, and we were kind of hinting at this too, there have been independent losses of tails in evolution since then. Yeah. And so this is this is kind of that blinking red light for the evolutionary biologist, is when you see something happening independently more than once, sort of lightning striking twice for genetics. And so there's convergent loss of tails in some prosimians. So some lorises don't have tails. Does that also involve TBXT? The authors don't say. I don't know how far they've kind of looked into that. There's also actually like a really important, I think, observation here is that in some primates, they've lost tails, including some macaques. So there are some macaques. So these are old world monkeys or African monkeys that have. There's some species that have tails and others that don't, and they're really closely related. And so what that implies is that tail loss happened sort of yesterday, evolutionarily speaking, not 30 million years ago, but within the last 100,000 years or 500,000 years or something like that. And so that's like a great comparison to draw, because there's so few other genetic changes that you might actually be able to not only see one big consequential change like TBXT, but also just a handful of changes. You might be able to sort of build that story a little bit more closely.
So I was just thinking now is maybe in a vertical orientation, bipedal, maybe the tail, which is connected to the spinal cord, right? It's all one series of bones. Maybe it makes too much of a down pull on the spinal cord. It's just not optimal for walking like that.
Yeah, and there's lots of labs that study this. By the way, I think it's like an important or a long-lived topic in anthropology and
of course.
And so there's a lot known about that. I just am not familiar with it. Yeah, but how much weight to put in on some of these things.
We have a few comments and questions. Should we?
Oh yeah, fantastic. I'd love to.
We have a couple of people who from places we didn't see before. Sue is from Tucson.
Great.
Kanami is from Kyrgyzstan. It's cool. Central Asian representation. Oh, and G. Ferraro says, so I had a tail. No.
Our ancestors did.
Our ancestors, right?
Yeah. If we can go to a time machine back about 30 million years, we'll see our great, multiply that by a lot, grandparents with tails.
And Andrew has a joke. Why did the skeleton refuse to sit down?
Uh-oh. Do you know?
Because he was cocksicky about his rear end support.
Oof, that's a deep track right there.
That's pretty
The
nerdy, right?
coccyx, yeah. The coccyx, I think, is what we call that. Those 3 to 5 vertebrae, the tailbone.
Sue says bobcats are tailless big cats. Yeah.
Yeah, yeah. Similar to the Manx cats, domesticated cats, all kinds of convergent evolution here. Independent events. You, as a evolutionary geneticist, you love to try to lift those up and draw comparisons here as you look for shared or different routes, genetic routes, to some of these phenotypes.
Pete says bipedal dinosaurs seem to have an important tail. Yeah, it's a big role in balance, right? Yeah.
Correct. And that's why, I'm trying to be a little bit careful about not overfitting some of the, the ideas about what was really acting as the selective pressure in the case of the tail loss, because
of course,
that's right.
There's some dinosaurs where they think they had spikes at the end of the tail and they were used in a defense, right? So that could even have another role.
Yeah,
besides balance. Yeah.
So it's a pretty safe bet that there's a lot of predator prey sort of outcomes, right? Because that's a really strong selective pressure. Are you able to eat and gain energy? If so, you'll have kids. Are you able to avoid getting eaten by something? If you're able to avoid that, you're much more likely to have kids. And so predator prey has driven so much evolution over the millions of years.
So Andrew says 6 fingers is better than 5, as base 6 maths means fewer irrational fractions than base 5. Okay.
That's right. The Hemingway's cats are onto something. The polydactyly is an extra finger trait is a good idea.
Abdulaziz, thank you so much for your contribution to science education, building spaces safe for science.
Celebrating science. And good to see you again, Abdulaziz. Really appreciate it.
So Andrew says, I'm beginning to think that evolution's random mutations are not so random. No, they are random, right, Nels?
Yeah, in this case, there's no reason to go outside of the sort of Darwinian boundaries, I think. The propagation of these selfish genetic elements, pretty random. I mean, there is, so the genome that the genetic sequences are being written on isn't completely random space. And so how selection acts on sort of the propagation of these things, there's certainly some tinkering around the edges here, but I think it's worthwhile. Depending on it's again kind of how the language of how we describe this, but at least as a first approximation, there's a lot of randomness in this process, and selection is really the thing that brings sort of directional energy to the outcomes.
So then Tom says the early hominids with this genetic change, so the ALU, would be heterozygous and then selection.
Yes, and so that's kind of an interesting point. So absolutely. So the very first one, the first jump would of an ALU, of that ancient ALU that we're seeing here today, would be a single event. But it's okay in the human case for this to become homozygous because it's not a deletion of exon 6. It's a probability that because of that other sequence that you'll remove exon 6. And so, one prediction would be if you're a heterozygote, that would be a more subtle phenotype. The homozygote, meaning you have 2 copies of that ALU, one on each gene, you'd still have that full-length copy, so it wouldn't be lethal, but that might be closer to, more of a genetic impact that then would influence the development towards the loss of the tail. Yep.
But in mice, the homozygous deletion is lethal.
Correct. And so, if they kind of tried, I think they really were hoping to genocopy like directly the human case to get 2 copies. So if they would have gotten 2 copies of the ALU, and then the second one nearby, that reverse complementing sequence, in the exact orientation, the prediction would be that even homozygous mice would be fine, but that they would have that tail phenotype. So that wouldn't be lethal. It's different than deleting out exon 6, where there's now, in the homozygous state, there's 0 full-length TBXTs. And it gets complicated. And I think, describing the genetics there and the genetic gymnastics, it's hard to sort of convey that. Yeah, it's a really good question.
And so someone else says, if it happened in a germline cell, this has
Yeah.
to be in the germline, otherwise it's not passed on, right?
Exactly right. Yeah. So the way, if this was just happening, so the alternative would be it's in a somatic cell. It's in like a skin cell, or in an adult cell. The reason it has to happen in a germ cell is both for it to be transmitted to the next generation, but also it has to step through that really early developmental window when the tail starts to develop or doesn't develop or develops in a shorter sort of configuration. Yeah.
So now let's summarize. From
Yeah.
we have 25 million years ago, we have our ancestors, monkeys with old world monkeys with tails, and then on a molecular basis, what happens then?
Yeah, so this random, so in the gene, like there are all of these of our ancestors running around, and in some subset of that population, even as much as like at the individual level, um, there is an ALU element that's hitchhiking off of a autonomous line one element. It jumps into intron 7 or intron 6, I guess, of between exon 6 and 7,
Right.
and then as a heterozygous event, somehow, and this starts to propagate to the point where you come back 30 million years later, and every single hominoid, from humans to gibbons, has two copies of this ALU in that genetic region. So this is this is like this improbability of these single, or very low probability events then propagating or being selected on over really long amounts of time, or you come back and it's actually quote unquote swept the population. All of our modern sort of, cousins have this thing.
So the idea would be that it's initially present on one gene copy, and it gives rise to a spliced mRNA, not all the time, but some of the time, which is lacking an exon, a piece of the protein. And then that causes a change in tail length. And it doesn't have to be gone. The experiments in mice show that gene can affect tail length severely, right?
Mm-hmm.
But it didn't have, those monkeys didn't have to lose their tail. It got a little shorter, and maybe that gave them some advantage. So from one individual, it propagated, and that takes many, many years to do that, thousands and thousands of years. But
Correct.
Then you have accumulating changes that somehow give an advantage, and we just don't know what it is because we're not sure. But Voitis says, is there any research if there was a change in pelvis structure after the loss of the tail? I don't know, but I'm guessing there is, right, Nels?
I'm sure there is. Outside so, and that gets to, this is not this is a point I wanted to make, actually. So after this paper was published, a little bit of a dust up on social media. So there's other scientists who I think do study exactly these kind of questions, who are a little bit like, hold on, you all are, you kind of genome scientists are a little bit new to this research area. And even in the language and how I think they describe some of the biology of tail, loss, or the presence of a tail bone, or reduction, or retraction, I think is an important phrase here. I don't know how it all plays out. Kind of was like fingernails on the chalkboard for some in the field that really think about development. So Kaz kind of, then it's sort of the classic social media scenario where authors feel like they were using insulting language, calling the authors naive, etc. The sort of people in the field are like, who do you think you are showing up in our field without knowing kind of what you're talking about? And so, the truth is probably somewhere in between. There's a lot of subtleties and nuance to this complexity. But you're absolutely right, Void. There's all kinds of complicated biology here. And so, so I think that's sort of the, as we think about this kind of through our podcasting lens, the lens of TWiEVO, it's where we're really interested is how can we use these new sort of genetic resources tools to understand a little bit more of our evolution. But that can really, hit some sort of cultural barriers, even among scientists, where, the history of the field or the way that science advances and how, studies might be made with argument or other forms or morphology. It's sort of an awkward collision between fields. And the reason we really want to lift up this kind of science and celebrate or talk about it is to just like let's do a little bit of the sociology here as well, to really I think ultimately these collisions are good and it's okay, and people can disagree or maybe you didn't say it the right way, etc. But have we learned something new that might be useful for understanding the mechanisms of evolution? I think the answer is yes. And so will they have every detail right? No. But will this sort of move science forward? I think the answer here is yes. And so, yeah, sorry. I mean, you're asking a really good, simple question, which I don't know the answer to. And so in our usual podcasting form, we just start rambling about bigger issues or points.
We have B from Algeria. Welcome.
Wonderful.
And here's the reason. Here's the selection, Nels. The obvious is the leaving of trees, more primates caught by the tail on the savannah.
There you go. I like that. Yeah, great predator-prey hypothesis here. So you're out of the trees, and you got to move fast, and you don't want handles for your predators to just
grab it. Yep. Here's our lunch.
Yeah, I like it.
Right.
Yeah,
cool.
Good call. Yeah,
I just love that paper. I think it's so cool.
Isn't it great? I mean, well written. Maybe a little fingernails on the chalkboard for some of the folks who think about the evolution of tails. But I think just really well done. The collision with the retro element, they had me at the ALU, right? The sine element.
It's great.
I can't get enough of that.
I wonder how long it took them to spot that, right?
Yeah. And so that's again, that gets to, the sort of the candidate gene search, narrowing it down, the sort of limited window. You can't do it. And so that's sort of the art of the science here is to, and what are the other contributions? I think this can help to maybe open some windows or doors into that as well.
So Sue asks, could the more recent tail loss in monkeys have spread more quickly through sexual selection?
Yeah, absolutely. I think that's exactly right. I mean, so there's the two of the really strong forces in evolutionary selection. One is what we've been talking about, predator prey, and that can extend into infectious microbes. By the way, you can think of those it's a little bit of a stretch, but that kind of falls a little bit into that kind of conflict zone. Yeah. And then the other one is sexual selection, as you're pointing out, Sue. And so the who you pick as a mate, or which so as we're thinking about mammals, which of the male germ cells, which of the sperm fertilize the egg? There's a lot of competition involved in that. So all the way from the kind of biological levels out to the behavioral levels of mate choice and things like that, with a lot of cheaters along the way, right? As we think about sort of species that take advantage of others in order to get them to protect cuckoo birds that will kick like the eggs out of the nest and then and sub in their own, and then they have sort of surrogate parents. So yeah, these are those are two of the biggest forces out there, and probably some combination of all of this, which again makes it pretty complicated. The tail is a really conspicuous feature, and so you can imagine that going into mate selection, sort of like the feathers of a peacock, like that's even in the same location. The tail could have a little bit of that energy.
Right. All right, Nels, should we move on to some picks here?
Yeah, and actually we should start with your pick, Vincent, because it'll keep our conversation alive here, I think.
Yeah, so I have a science paper, and I'm going to put it up here so you can see it. This is a paper from the Sultan Qaboos University Medical Journal, a true human tail in a neonate. It's a case report. So here it is. A true human tail is a benign vestigial caudal cutaneous structure. Okay? Yep. It can be a true tail can be distinguished from a pseudotail, as the latter is commonly associated with underlying spinal disraphism, which requires specialized management. True human tails are very rare, with fewer than 40 cases reported to date. So there have been 40 people reported who have true human tails. And what they're reporting here is a healthy one-day-old male, newborn, referred to this hospital in India in 2014 with a cutaneous appendage arising from the lumbosacral region.
Wow. Yeah.
MRI of the spine ruled out spinal disraphism. The appendage was removed by simple surgical excision. And they say clinicians should emphasize use of true tail and pseudotail as specific disparate terms as the clinical, radiological, and histological findings differ. So they have a picture here, all right? This is a newborn. It's just amazing. Look.
Yeah, the tail.
Clinical photograph of a male newborn showing a cutaneous. Look at this. Oh my gosh. It's a tail. It's a true tail, right?
Yep.
And this happens very rarely. 40 is nothing in terms of human birth, right?
Super rare.
Oh my gosh. That's amazing, right?
It's amazing. Yeah. So as a geneticist, so we've talked a little bit about this already, Vincent, that, how do you study something in humans? So this could be an opportunity if in any of these 40 cases, some of that tail tissue or just any tissue, a blood sample, whatever, was recovered from those patients.
Yes.
Right? So what you would do is you would sequence the TBXT gene. Is that ALU element missing? Would be a very direct hypothesis that builds out of the study that we've been talking about. Or are there other changes or like rare changes, right? That's how you might sort of really connect this directly to the human case in these super rare occasions where you see a tail. Now, there's also, again, you have to be careful, I think, from an evolutionary standpoint. So, is this a sort of a new, I mean, maybe even in a more exciting way, is the sort of return of the tail, is that because you've reversed sort of the genetic in the first place, or have you, in a new mutation, have you rewritten how development happens to recreate a tail? Those are actually two different things, where it's the loss, it's sort of like the loss of a thing, blocking the loss of a thing versus reintroducing it through another genetic pathway. And so really tough to study because as you were saying, like these cases are super rare, but there's that possibility, even in human sort of diversity, that there could be there could be tails out there.
I think that's a good explanation of how mouse results can then be tested in humans by, in this case, simply sequencing the gene, right? You could see what's going on there.
That's right.
That's very cool.
Yep.
Now, the reason I looked up this paper is because when I was a postdoc in 1980, 79, 80, one of the other postdocs, Fred Ledley, in the lab, had, he was an MD, was famous for having taken care of a baby with a tail appendage, and we were all just flabbergasted. We didn't know that happened. And I'm curious about if you leave this tail on, right?
Yeah.
What happens to it? Does it remain 6 inches long or whatever? Does it get 5 feet long? Does it drag on the floor? I mean, that's an experiment you don't want to do because you want to make a normal human, right?
Yeah. Yep.
But it's an interesting question.
It's an interesting question. And it also, I think it just points to how, like, in one sense, the tail can be almost, or the tailbone is just sort of vestigial structure. But when we change, as humans, when we change our form, our morphology, if you have it really grabs our attention somehow. It kind of hits you in this visceral, way. And so that kind of illustrates that something about our, how we identify ourselves as humans. It really cuts to the core.
We are self-aware, Nels.
Some of us more than others.
Well, the reason I say that is because we did a paper on immune where they find that mice are self-aware.
Yeah, interesting.
If you paint a mark on their forehead and you put them in front of a mirror, they will actually start to put their paw on it because they can see that's not me. What's going on here?
Yeah, interesting. I love it. Yep.
Isn't that cool? What do you have for us, Nels?
Yeah, so my pick of the week isn't quite as controversial in some, well, maybe it is in different ways, but so will brie cheese go extinct? So this is back to the microbes again, so microbiology here. So the fungi, the strains of fungi that go into the sort of cheese industrial complex, humans have changed how cheeses would sort of, in the early days of humans messing around and creating cheeses, we've obviously put our sort of technological stamp on this. And so now all of the fungi that's used in cheese making for brie and camembert cheeses is just a single strain, Penicillium camemberti, can't reproduce on its own. And so we clone it again and again, taken out some of the sort of diversity and the complexity of the microbial constituents, the fungi, in cheese making. And so this means that every cheese is made with a genetically, in these days, identical strain. The lack of genetic diversity makes it vulnerable to pathogens and other environmental changes. And so I love this because it's sort of the Red Queen hypothesis, the idea that having genetic diversity is what sort of allows lineage of species to exist. This could be making our cheeses more vulnerable if these clonal strains get attacked by pathogens or go extinct. And so my link is to a Washington Post story that was covering this idea. It's a different paper out there. And the good news is we're probably okay. Brie cheeses will go on. In worst case scenario, if the industrial strains that we use today are lost, other fungi are in the wings waiting to take over and maybe even contributing to the diverse development of cheeses into the future.
Cool. Yeah, we had a similar paper on TWiEVO a while ago about blue cheese, where there are apparently 4 different strains of whatever it is that you use to ferment blue cheese, and they're having trouble with them. So same idea. We don't want to lose our beloved cheeses, right?
No, we need cheese. That's if we can agree on one thing, hopefully it's that.
So someone commented at the end of that article, I long for a world where cheese is the only problem.
Yeah, there you go. 99 problems, but cheese ain't one.
All right. Well, Nels, I think it's time to wrap up, don't you?
I do. This has been really fun.
Yeah, it's a good paper. I really like that. It's a good one for the next 100 of TWiEVO.
That's right.
And that's 100. That's like 2 years from now, we're going to hit 200, right?
Yeah, or at our current pace, it would be another almost 8 years, I think. 7 years.
Oh, that's
maybe.
right. I'm thinking of weeks instead of months.
Yeah, but we'll be here, TWiEVO 200. Get your tickets now for our in vivo event.
Wherever that may be. Well, you can find TWiEVO at microbe.tv/TWiEVO. If you want to send in some questions or pics, you can send them to TWiEVO at microbe.tv. And remember, if you enjoy this work that we do to make science spaces safe for science, we'd love your support to do this. We'd like Microbe TV to grow and encompass more people and more ideas, and we want your help to be able to do that. So you can go over to microbe.tv/contribute to do that. Nels LD is at selvolution.org. On Twitter, he's L Earlybird. Thanks, Nels.
Yeah, thank you. And thanks to all our listeners with great questions here as well. Really, that's another way to participate in TWiEVO is through the discussion. Yeah. I really appreciate it.
I'm Vincent Racaniello. You can find me right here at microbe.tv. I want to thank our moderators today. We had Barb Mack, we had Andrew, we had Vanity Nutrition, we had Tom, and we had Les. And there are 53 of you right now and 34 likes. Hit the like button and get us up closer to 50, okay? That would be nice. Just helps people find us. And I want to remind you that in less than 6 hours from now, we will have office hours tonight at this place, at the same place where you found this, a slightly different link. But I will be there. I'm going to try and talk a little bit about H5N1 influenza virus.
Oh, yeah.
I did have a mini lecture that I used to give. I'm going to try and find it somewhere on one of my hard drives. And if I can do that, we'll talk about that.
Yeah, and is H151, is this the one that's showing up in some cattle, including in Texas?
It showed up in some cattle, and apparently a human got it in Texas. But it showed up in cattle in a number of states. It doesn't kill the cattle, but it makes them make less milk, and you don't want to distribute that milk. But there's been one person in Texas who apparently got it from a cow. So whether that's concerning or not, we can talk about. That's 8 p.m. Eastern, right here on YouTube. The music you hear on Trampled by Turtles, no, the music you hear on TWiEVO is by Trampled by Turtles. You can find their work at trampledbyturtles.com. Let's start the music here.
Yeah.
You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, stay curious. Bye-bye.